This video will cover question 52 of the Specimen Section 1 PBSAA paper. Please note that our solution will be the same as the equivalent Enter Specimen Section 1, question 69. In question 69, we're asked which of four statements about the genetic modification of a bacterium to produce human insulin are correct. Well, before we have a look at those four statements, let's remind ourselves of this process. We can generate human insulin from bacterium by first extracting the insulin gene from the human nucleus, using specific restriction enzymes. So once we've extracted those strands, we can place them into bacterial plasmids, which are single circular loops of DNA present in the bacterial cytoplasm, and then following transcription and translation at the ribosomes, we can then generate our protein product, insulin. So we are changing the genome by inserting human DNA into bacterial plasmids. That's how we get human insulin from a bacterium. Let's have a look at our four statements. Firstly, human insulin DNA is cut out using an enzyme. When we know this to be true, it's a specific restriction enzyme. Restriction enzymes cut DNA at specific sites. Moving on to 2, the DNA is inserted into the nucleus of the bacterium. Well, as we've highlighted, there is no nucleus in a bacterium. They have no true nucleus, they just have a single cytoplasmic region of DNA, or a number of them, but one main source of most of their DNA, but which does not have a membrane-bound nucleus as eukaryotic cells do. Moving on to 3, the modified bacterium is cultured. Well, yes, the modified bacterium has to be cultured so that we identify the bacterium which happen to have the insulin gene inserted, because not all of them will have the successful procedure carried out. And then, once we've cultured them, we can increase our yield of insulin, because one bacterium takes quite a while to produce any useful amount of insulin. And finally, 4, the bacterial cells are injected into humans with diabetes. Well, by and large, it's not a good idea to ever inject bacterial cells into human beings, whether they have diabetes or not. This represents a misunderstanding of what this process involves. What we are doing is extracting insulin, which can then be given to diabetic patients as medicine, since they can't produce insulin themselves. So our answer is B, that 1 and 3 are our correct statements.